A hollow tube of ice can grow downward beneath the ocean, surrounding a stream of sinking concentrated brine.
The process begins because winter air cools the sea surface far below the temperature of underlying ocean water.
When seawater freezes, water molecules join the ice lattice, while most dissolved salts remain outside the crystals.
Rejected salt concentrates the remaining liquid, producing brine that is colder and saltier than ordinary seawater during channels. Young sea ice is not a solid slab. It behaves like a porous mixture of ice and concentrated liquid. As cooling continues, connected pathways allow increasingly dense brine to drain through the sea ice under gravity. Additional dissolved salt increases density, making the rejected brine heavier than the surrounding ocean water below during ice.
Once released, the cold brine begins sinking as a narrow gravitational plume beneath the floating sea ice. High salinity lowers the brine's freezing point, preventing the descending core from solidifying immediately. Crystallization builds growth below.
The surrounding ocean can freeze when the plume cools it below the local seawater freezing temperature during sediment.
Ice, therefore, forms around the brine stream, creating the first thin wall of a hollow structure during winter. Continued discharge extends the sheath downward because fresh seawater freezes near the moving plume tip. Freezing acts naturally. The brinicle grows from its lower edge rather than sliding downward as one completed piece of ice. Its final shape resembles an inverted chimney carrying a dense cold liquid downward instead of hot gas upward. Both structures organize flow within a solid wall, but temperature and gravity reverse the familiar direction during tank. A true brinicle is hollow, with an icy shell continuously surrounding a liquid brine core. Gravity acts seasonally. Brine flow controls how quickly the tube lengthens and how thick its walls become during formation, during plume. Too little flow cannot sustain growth, while excessive flow may destabilize the forming tube and surrounding water.
Stable brinicles require a narrow combination of cold discharge, favorable salinity, and relatively gentle surrounding currents during ice. The visible ice finger is the successful outcome of many hidden processes occurring within the sea ice above. Brine rejection begins at the microscopic boundary where ordered ice excludes most impurities from its structure during seafloor. Cooling reduces liquid volume and forces salts into increasingly concentrated brine inclusions within the ice. Dilution acts efficiently. Brine chemistry evolves during cooling because individual salts have different solubilities and precipitation temperatures. Flow stabilizes branching below.
Salt composition influences freezing point, viscosity, density, and the shape of any tube that later develops during seawater.
Temperature and salinity determine whether the mushy layer remains permeable enough for gravity drainage. Discharge determines growth below. If channels lose connectivity, concentrated brine remains trapped and cannot feed a sustained downward plume. Drainage acts structurally.
Small thermal changes can reopen pathways and release stored brine in sudden drainage events. Pressure alters stability below. Converging drainage concentrates enough cold brine at one point to support a visible brinicle. Brine maintains persistence below.
The outlet geometry helps anchor the tube while the plume begins freezing nearby seawater. Crystallization builds transport below. Reinforced upper connection supports the growing structure against its own weight and weak surrounding currents during Antarctica. Brinicle growth continues only while the overlying ice supplies sufficiently cold, concentrated brine through connected channels during ice.
When the brine source weakens, the lower growth front slows and eventually stops extending. Freezing records freezing below. An inactive tube may flood, fracture, or melt after its internal brine circulation ends. Salinity shapes exchange below. Ambient flow can bend fragile brinicles or detach them before they reach the seafloor. Transport connects flow below.
Many short-lived tubes probably disappear without ever being observed by cameras or divers. Heat flow governs formation below. Low turbulence allows a delicate ice wall to survive long enough for continued downward growth. Gravity acts repeatedly.
Crossflow tilts the descending brine and forces the ice tube to grow along a curved trajectory during interface.
Natural brinicles can twist, branch, or lean because real polar seawater is never completely motionless. Turbulence acts continuously.
Changes in plume structure can create multiple outlets and complex frozen geometries beneath the same source during tank.
Despite irregular exteriors, the defining feature remains a liquid pathway enclosed by newly frozen seawater. Cooling acts thermally.
Brinicle is a tube, not a solid spike, and its interior remains actively connected to the sea ice. Heat flows toward the supercold core, allowing seawater beside the plume to release latent heat and freeze. Every new layer of ice releases latent heat that must be carried away by the surrounding water and brine. Growth rate depends on whether nearby water can remove that released heat efficiently. Density steadily drives persistence below. Warm surroundings or weak heat removal reduce freezing and produce thinner, less stable structures. Discharge determines transport below. Extremely favorable thermal conditions can freeze a wall quickly before the plume travels far downward. Drainage acts seasonally.
Wall growth can eventually choke its own supply, creating a self-limiting feedback within the tube during winter. Continued flow helps preserve the hollow center by delivering saltier liquid with a lower freezing point during seawater. The lower opening must remain unsealed so the dense brine can continue sinking beyond the ice wall. The frozen tube records the path of a cold descending wake that continues beneath its visible endpoint. Entrainment dilutes the plume, reducing density, salinity, and the ability to freeze additional water. Mixing weakens formation below. Only the colder concentrated case remains below the surrounding seawater's local freezing threshold. Freezing steadily records geometry below. Combined thermal and salt conditions decide whether the plume sinks, mixes, freezes nearby water, or disperses into the ocean. Freezing around the plume can concentrate its remaining liquid further, reinforcing density and downward motion. Transport acts gradually. Brinicle formation participates in a small local circulation driven by both temperature and salinity differences. Heat flow acts thermally. Surrounding seawater continually replaces liquid frozen into the expanding ice wall. Gravity steadily guides local structure below. Sediment and plankton can make local flow patterns visible around an actively growing brinicle. Chemistry modifies growth below. The most extreme temperature and salinity conditions remain concentrated near the liquid core and lower outlet during sediment. Brinicles range from small, fragile tubes to structures capable of extending several meters beneath sea ice during winter. A sufficiently persistent source can carry the frozen chimney all the way toward the ocean bottom during seawater. When Brinicle reaches bottom, its dense liquid core continues flowing outward across the seafloor. Ice growth acts seasonally. The cold brine spreads as a gravity current because it remains denser than the water above it. Bottom topography guides the plume into channels, hollows, and low points across the polar seabed. Flow acts chemically. Nearby seawater can freeze along the spreading plume when local temperature falls below its freezing point during channels. Horizontal freezing may extend beyond the Brinicle base, while the dense liquid continues moving inside and beneath it. Slow benthic animals may struggle to leave a rapidly cooling patch when the plume reaches their habitat. Biological outcomes depend on mobility, exposure time, body size, and the precise path of the dense flow. Documented imagery can appear dramatic, but a brinicle does not instantly freeze every organism it encounters during shell. Organisms may face freezing, osmotic stress, or simple displacement, depending on their location relative to the plume. Hypersaline brine draws water from tissues and can challenge animals even where new ice never touches them. Sea ice brines also contain microorganisms adapted to cold, salt, and rapidly changing chemical conditions. Drainage can transport dissolved chemicals and microscopic life from the ice into the ocean below. Freezing acts naturally. A brinicle is therefore part of a broader exchange pathway between sea ice and seawater. Salinity acts briefly. Dilution eventually weakens the extreme salinity and temperature that sustain bottom freezing. Transport steadily connects local mixing below.
After discharge declines, the remaining chimney may persist briefly as an inactive frozen structure. Heat flow governs stability below.
Motion between floating ice and fixed seafloor can bend or break a grounded Brinicle. Gravity guides persistence below. Attached pieces disappear when surrounding conditions no longer support their unusually cold frozen walls. Chemistry modifies transport below.
Brinicle formation is episodic, following where drainage channels open and kloof beneath evolving winter ice. Turbulence acts chemically.
The phenomenon is most likely during active sea ice growth, when strong cooling drives concentrated brine rejection. To isolate these interacting controls, researchers recreated Brinicle growth inside carefully monitored laboratory tanks. Cooling controls freezing below. Laboratory experiments reproduce Brinicles by injecting dense, cold brine into water with a higher freezing point during seafloor.
The artificial tube confirms that a descending plume alone can organize the essential hollow structure. Dilution acts efficiently. Sensors reveal how temperature gradients evolve as the wall grows and the plume entrains surrounding water during winter. Separate measurements distinguish concentrated brine, newly frozen water, and unchanged ambient liquid outside the tube. Density acts gradually. Adjusting discharge rate directly changes tube length, diameter, stability, and the speed of downward growth. Discharge acts thermally. Weak flow cannot maintain the thermal plume needed to keep extending the hollow ice front. Drainage acts structurally.
Controlled crossflow demonstrates why natural currents can determine whether a Brinicle survives or collapses. Pressure alters branching below. Different salts produce distinct growth rates, wall textures, and branching patterns under otherwise similar conditions. Brine acts briefly. Brinicle shape contains information about the chemistry and dynamics of the liquid flowing inside it. Crystallization acts steadily. Despite varied shapes, all successful laboratory examples follow the same basic thermodynamic and gravitational process. Geometry acts locally.
Brinicles resemble chemical gardens because a flowing solution builds a solid tubular wall around itself. Mixing acts seasonally.
Gravity, density, and temperature reverse the direction while preserving the self-organized hollow geometry. Freezing records persistence below.
One system deposits minerals, whereas the polar version freezes surrounding water into an icy membrane. Salinity acts chemically. Sustained internal flow allows both structures to extend far beyond the point where formation originally began, during sediment. Brinicle walls are active boundaries where heat transfer, salt exclusion, and fluid motion remain tightly coupled during winter.
The frozen shell is not perfectly pure or impermeable, especially during rapid laboratory growth. Gravity guides freezing below. Low wall defects can redirect brine and generate branching or double-walled morphologies. Chemistry steadily modifies exchange below.
Competing channels may merge when their thermal fields overlap within the same surrounding water. Turbulence limits flow below. Computer models test how coupled fluid dynamics and freezing reproduce the laboratory structures. Convection steadily redistributes formation below. The same physical framework can be explored under conditions impossible to reproduce directly on Earth. Cooling acts thermally. Scientists have proposed that brine drainage beneath ocean-world ice could create structures analogous to terrestrial brinicles. Freezing ocean water would reject dissolved salts and concentrate liquid brine within the growing shell. Dilution acts naturally. If pathways remain connected, dense brine could descend toward Europa's ocean beneath the ice. Flow stabilizes branching below. No spacecraft has yet observed a Brinicle beneath Europa's inaccessible ice-ocean boundary. Density steadily drives structure below. Different gravity, pressure, and composition could produce structures unlike anything seen beneath Antarctic sea ice. Discharge acts locally. Models suggest thicker shells may permit larger, more persistent drainage features at the ice-ocean interface during sediment. Such drainage could move chemically important materials between an icy shell and its hidden ocean. Pressure acts dynamically. Transported compounds might create chemical gradients, although biological consequences remain entirely speculative. Enceladus presents another possible setting where freezing salty water could concentrate and drain brine. Crystallization builds transport below. Hypothetical brinicles should not be confused with the vapor and ice jets already observed escaping Enceladus during ocean. Shared physical principles may operate across worlds while producing very different structures and chemical behavior. Mixing acts repeatedly.
Earth remains the test case for understanding how cold brine self-organizes beneath floating ice. Freezing acts efficiently.
Improved field sensors could capture the internal conditions of growing brinicles in real time. Salinity shapes exchange below. Simultaneous measurements would connect tube geometry with the invisible brine plume producing it. Transport steadily connects flow below.
Broader surveys could reveal how common brinicles are and which sea ice conditions favor their growth during ice.
Combining environmental maps may eventually allow researchers to predict where brinicles should form. Gravity steadily guides geometry below. Climate-driven changes to sea ice growth could alter brine rejection, though local brinicle responses remain uncertain. Brinicles illustrate local physics, but also belong to the broader desalination process occurring throughout growing sea ice.
Each stage is understandable alone, yet together they build one of the ocean's strangest self-organized structures.
A brinicle grows downward because dense liquid falls first, while freezing seawater constructs a hollow chimney around it.
This small lizard escapes predators by diving underwater and repeatedly breathing from a silver bubble attached to its snout.
Water anoles inhabit tropical stream margins, where dense vegetation, wet rocks, and moving water provide immediate underwater refuges.
Their streamside habitat places branches, roots, and climbing surfaces directly above pools, channels, and concealed submerged shelter.
Perching close to water lets the animal disappear from danger within seconds, before many terrestrial hunters can react.
Birds, snakes, and other predators create intense pressure for rapid escape behavior along exposed rainforest stream banks. Before diving, the lizard evaluates distance, cover, current, and the nearest safe route beneath the water safely.
A sudden leap carries the animal below the surface before most land-based predators can follow or accurately track it.
Once submerged, depth, darkness, ripples, and visual distortion make the small reptile surprisingly difficult to locate from above.
Strong toes and curved claws help it remain anchored against flowing water while hiding beneath rocks or roots.
The extraordinary respiratory behavior begins when air leaves the nostrils and forms a bubble instead of floating away. Hydrophobic scales help exhaled air remain connected to the face, keeping the bubble accessible beside both nostrils. Semi-aquatic anoles possess scale features associated with stronger water repellency and more reliable underwater bubble retention. The nasal bubble may connect with a broader reflective air layer trapped against hydrophobic scales across the head.
Air moves through the nostrils while the mouth stays closed, preserving a compact respiratory circuit beneath the surface.
The lizard inhales from the bubble, exhales back into it, then repeats the same cycle during submersion.
Every breathing cycle changes the bubble's composition because metabolism consumes oxygen while tissues continuously produce carbon dioxide.
Oxygen follows the ordinary vertebrate route from the lungs across thin tissue and into the circulating bloodstream.
Rebreathing extends the usefulness of existing lung air, rather than creating an entirely new respiratory organ underwater. Measured oxygen decline inside the bubble confirms that its trapped air actively supports the submerged animal's respiration. Repeated inhalation lets the lizard reuse air that would otherwise escape immediately toward the stream surface after exhalation. Recycling stretches the useful duration of a limited air store, while the animal remains concealed during a defensive dive. Each extra underwater minute increases the chance that a searching predator leaves before the lizard must resurface. Staying below the surface allows currents, shadows, and ripples to erase the original location of the sudden escape. Recorded submersions vary greatly because body size, stress, temperature, habitat structure, and individual decisions all influence duration. Some semi-aquatic anoles have remained underwater for roughly 8 minutes, although most defensive dives are considerably shorter. A maximum observation reveals biological capacity, not the routine dive length of every animal under natural conditions.
Body size, water temperature, current speed, and disturbance can all change how long a submerged lizard remains hidden.
Smaller and larger animals carry different lung volumes, metabolic demands, and bubble surface areas relative to body mass. Remaining nearly motionless lowers oxygen consumption, allowing the bubble's limited respiratory value to last substantially longer under threat. Sheltered positions behind roots and stones reduce physical effort while hiding the lizard from movement above the water. Calm pockets behind obstacles demand less energy than exposed current, where the animal must constantly resist downstream force.
Moderate flow can refresh gases around the bubble without tearing the delicate air layer away from the snout. Carbon dioxide readily dissolves into water, creating a possible route for respiratory waste to leave the bubble. Because carbon dioxide is highly soluble, surrounding water may absorb it faster than oxygen disappears from the reservoir. An exposed bubble water boundary could therefore remain useful longer than a completely sealed pocket containing the same air.
Metabolic carbon dioxide travels from active tissues through blood toward the lungs, even while the lizard remains submerged. Exhaled carbon dioxide enters the nasal bubble, where some molecules may diffuse outward into the passing stream. Carbon dioxide removal is physiologically plausible, but recycled stored air remains the mechanism supported most directly by experiments.
Underwater rebreathing combines ordinary lungs, hydrophobic skin, trapped air, and a dynamic gas boundary beside the nostrils. The respiratory loop begins with oxygen already stored inside air, carried beneath the surface during the initial dive.
Bubble oxygen concentration falls predictably while a living lizard continues breathing repeatedly from the attached reservoir underwater.
Researchers compared that decline with artificial bubbles held underwater under similar physical conditions, but without breathing animals.
Control bubbles retained oxygen more consistently, demonstrating that biological respiration caused the stronger decline seen with lizards. Those measurements confirmed that the nasal bubble functions as an extension of the animal's existing pulmonary air supply. Oxygen enters the lungs during inhalation, while mostly unused nitrogen returns to the bubble during the following exhalation. As oxygen decreases, nitrogen occupies a greater fraction of the remaining bubble, and the respiratory mixture becomes progressively poorer.
Eventually, available oxygen falls below a useful level and contributes to the lizard's decision to return toward air. The animal releases its underwater grip, swims upward, and breaks the surface to obtain a completely fresh breath.
At the surface, the depleted bubble merges with open air, while the lungs refill within a single respiratory cycle. After climbing onto a nearby rock, the lizard restores normal oxygen delivery while monitoring the streamside environment for danger. Heart rate, breathing, and movement gradually return toward ordinary levels as the animal recovers between defensive dives.
Another predator may appear before recovery finishes, forcing the lizard to repeat its underwater escape under greater physiological pressure.
A second dive creates another nasal bubble immediately, showing that the behavior is repeatable whenever danger returns.
Every surfacing restores a fresh gas mixture, replacing the oxygen-poor reservoir produced during the previous submerged interval. Across a single day, repeated cycles of diving, rebreathing, resurfacing, and recovery may occur around the same stream refuge.
Underwater movement may also support exploration or foraging, although predator avoidance remains the best established ecological function. Submerged stones, roots, and leaf pockets contain potential prey, as well as concealed resting places away from terrestrial hunters.
The lizard balances underwater opportunity against limited oxygen, physical effort, and the continuing need to return for atmospheric air.
Scientists tested the bubble's actual respiratory value by experimentally preventing its normal formation during controlled defensive dives.
Hatched lizards entered identical water chambers, differing mainly in whether the familiar nasal air bubble remained available.
Control animals preserved their ordinary hydrophobic air interface and continued cycling exhaled gas beside the nostrils underwater.
A temporary surface treatment disrupted bubble attachment without changing the chamber's water temperature, depth, or overall handling conditions. After treatment, exhaled bubbles detached from the face and floated upward before the lizard could inhale them again.
Those animals relied mainly on their initial lung supply, because reusable gas no longer remained beside the nostrils.
Lizards retaining normal bubbles stayed submerged substantially longer than animals whose external air interface had been disrupted. In that experiment, ordinary rebreathing increased average dive duration by approximately 32% under otherwise matched conditions.
Comparable body sizes, chambers, and handling procedures strengthen the conclusion that access to the bubble caused the difference.
The result demonstrated that the air pocket performs a functional respiratory role, rather than merely reflecting trapped surface air.
However, the experiment did not show permanent underwater breathing, unlimited oxygen renewal, or complete independence from atmospheric air.
The bubble remains a finite reservoir, whose volume and gas composition continue changing throughout every submerged breathing cycle. Cycling improves how the initial air supply is used, but it cannot prevent eventual oxygen depletion during long dives. Possible carbon dioxide loss across the bubble surface may further delay the point when resurfacing becomes physiologically necessary. Gas may move in both directions across the interface, although each proposed exchange process requires separate experimental evidence. Confirmed rebreathing, plausible carbon dioxide removal, and emerging oxygen uptake evidence therefore occupy different levels of scientific certainty. Successive studies progressed from behavioral observation toward gas measurement, bubble disruption, and controlled environmental experiments over several years. Natural dives remain essential because laboratory mechanisms evolved to solve ecological problems along predator-filled tropical streams.
In the wild, bubble-bearing lizards hide beneath ledges and roots, while threats continue searching along the bank above.
An extended dive allows danger to move farther away before the concealed animal cautiously returns to the surface. Comparative research revealed that underwater bubble rebreathing appeared independently in several distantly related semi-aquatic animal lineages. Separate evolutionary branches developed remarkably similar diving behavior, despite lacking one recent bubble-breathing common ancestor within Anolis. Streamside species from different American regions encountered comparable combinations of predators, water access, and underwater shelter near flowing water. Those recurring ecological pressures repeatedly favored escape strategies that turned nearby streams into temporary defensive refuges during attacks. Across independent lineages, threatened lizards entered water and formed accessible nasal bubbles using similar behavioral sequences under threat. Ordinary animal skin already possessed water-repellent microstructure before specialized underwater rebreathing evolved within semi-aquatic species. Natural selection likely co-opted that existing surface property, giving hydrophobic scales an additional respiratory function underwater. Water repellency may originally support self-cleaning or surface protection, while later becoming useful for stable bubble retention. Semi-aquatic forms maintain more persistent external air films that remain attached during movement beneath the water surface. The accessible air concentrates near the nostrils, allowing repeated inhalation while the lizard keeps its mouth closed underwater.
Different species vary in bubble size, attachment area, head shape, and the number of threads within their air layer.
Convergent solutions still reflect each species' body dimensions, scale texture, habitat, and preferred underwater hiding behavior underwater.
Local stream conditions can determine which bubble geometry and diving strategy produce the greatest survival advantage locally.
Fast rocky channels and slow shaded pools impose different costs through current strength, oxygen availability, and concealment. Calm water reduces the energy needed to hold position, though surrounding gases may refresh differently than in strong flow.
Water containing more dissolved oxygen creates stronger conditions for oxygen to move toward an attached underwater bubble.
Researchers therefore compared dive duration across chambers containing low, intermediate, and high dissolved oxygen concentrations in controlled trials.
Lizards remained submerged longest in the highest oxygen treatment, when water movement was kept relatively low and controlled.
Separate experiments manipulated dissolved oxygen and flow independently, preventing one environmental variable from masking the other's effect.
With oxygen held constant, slower flow treatments also produced longer dives than faster-moving water inside the experimental chambers.
The combination of adequate dissolved oxygen, low physical effort, and a stable bubble suggested an additional respiratory possibility.
A true physical gill would draw some dissolved oxygen from surrounding water into the bubble during submersion.
Aquatic spiders and insects already use air films or bubbles that exchange respiratory gases directly with water.
Across such interfaces, oxygen can enter from water, while carbon dioxide leaves through the same exposed boundary.
The animal bubble changes volume and shape during dives, potentially recording several simultaneous gas movements across time.
Closed reservoir and physical gill models predict different oxygen trajectories, bubble volumes, and achievable underwater durations for the animal. Longer dives in oxygen-rich water match one important prediction expected from partial physical gill activity underwater. However, researchers have not yet measured continuous oxygen flux directly across the bubble's surface during natural rebreathing. Future microsensors could monitor gas movement at the interface without disturbing the lizard's behavior or hydrophobic air layer.
Simultaneous oxygen, carbon dioxide, and volume measurements would reveal how storage, waste removal, and replenishment interact underwater.
Portable field sensors can connect controlled respiratory mechanisms with temperature, flow, and dissolved oxygen in occupied streams. Wild lizards frequently perch near stream sections offering manageable current, adequate oxygen, and nearby underwater cover during danger.
Roots, ledges, and cavities provide better concealment than open banks, even when respiratory conditions are otherwise similar.
Seasonal rainfall, sediment, temperature, and vegetation can alter both the quality of refuges and bubble performance throughout the year. Warmer streams hold less dissolved oxygen while simultaneously increasing the lizard's metabolic demand during underwater hiding at the same time. Shaded riparian forest may preserve cooler, oxygen-rich channels better than exposed streams altered by canopy loss. Bubble breathing, therefore, depends on an intact combination of stream structure, water chemistry, vegetation, and predator ecology. Repeated breathing, oxygen decline, bubble removal effects, and dissolved oxygen experiments now support several connected parts of the mechanism. Evidence is strongest for recycled air, while direct oxygen replenishment from water remains an active research question. A tiny nasal bubble helps this lizard stay hidden by combining stored air, gas exchange, and remarkable surface biology. Above powerful thunderstorms, enormous electrical flashes can appear where the atmosphere is already approaching the edge of space. These events occur far above ordinary weather, across atmospheric layers most people never associate with lightning. Above storms, conventional lightning moves through clouds or toward ground, but other electrical disturbances illuminate the sky overhead, across altitude. At those heights, air density, pressure, and electrical breakdown behave very differently from conditions near Earth's surface. Thin air allows enormous luminous structures to develop from surprisingly small amounts of excited gas, above active storms. Excited nitrogen produces much of the characteristic red light seen in high-altitude sprites. Structure identifies mesospheric response. Most transient luminous events are faint, brief, and easiest to detect with sensitive cameras after darkness falls. A complete event may unfold within milliseconds, disappearing before an observer can consciously process its shape. Across altitude, high-speed imaging transforms one mysterious flash into a sequence of rapidly developing electrical structures. During brief flashes, the storm below can trigger light tens of kilometers above its cloud tops, without a continuous visible bolt. Understanding the upper flash begins with how collisions inside thunderstorms separate electrical charge. This signal resolves storm charge. Violent collisions among ice and water particles help organize charge throughout a growing storm. Contrast tracks mesospheric response. Storm dynamics create large reservoirs of opposite charge separated across several kilometers of cloud. Boundary locates lightning coupling. As charge separation increases, the surrounding electric field strengthens throughout the cloud and nearby atmosphere during brief flashes. Lightning begins when electrical breakdown creates conductive channels through air that was previously insulating. Transition compares electrical breakdown.
Some sprite-producing flashes transfer positive charge from the cloud toward Earth in exceptionally powerful strokes. After lightning,
That rapidly changing field can push thin upper air beyond its local breakdown threshold. Emission exposes mesospheric response.
Sprites occupy the mesosphere and lower ionosphere, dramatically higher than thunderclouds or commercial aircraft, within thin air.
Their scale becomes clearer when placed above aircraft routes and below most orbiting spacecraft. Evidence frames sprite formation. A sprite can illuminate a region far larger than the storm feature that appears to trigger it. Enormous size does not imply long duration, because most visible sprite structure develops almost instantly, during brief flashes. Some sprites follow their parent stroke almost immediately, while others appear after a measurable delay, above active storms.
Both rapid charge removal and continuing current can influence whether the upper atmosphere reaches breakdown within thin air.
Researchers use charge moment change to describe how strongly lightning alters the electric field above a storm.
Large charge moment changes strongly increase the probability that a lightning flash produces a sprite across wide regions. Even powerful positive strokes do not guarantee sprites because atmospheric conductivity and storm geometry also matter within plasma. Sprite initiation depends on several interacting conditions rather than one simple lightning measurement. This boundary maps global occurrence. Nighttime conditions favor optical detection and change the electrical response of thin mesospheric air above active thunderstorms.
Solar radiation alters electron density, affecting how electric fields build and dissipate above storms. Contrast tracks charge transfer.
Many events are observed at night because their faint emissions would disappear against bright daylight across wide regions.
Observers often photograph sprites from distant clear skies rather than standing beneath the parent thunderstorm during brief flashes.
Distance can improve visibility by exposing the entire region above a storm's cloud shield. Transition compares global occurrence.
Sensitive imaging captures emissions too brief and dim for ordinary cameras under most conditions. Network supports upper fields.
Precise timing links each upper atmospheric event with candidate lightning activity inside the storm below, after powerful lightning. These matched observations establish the connection between sprites and powerful lightning charge transfers. This channel distinguishes storm geometry. Sprites occur worldwide wherever sufficiently energetic thunderstorms interact with favorable upper atmospheric conditions. They are not limited to tropical convection, although storm structures and triggering patterns can differ above active storms. Many sprites resemble luminous jellyfish, with a bright body suspended above long descending tendrils. Channel tests ionospheric change. Individual streamers propagate through localized electric fields and build the sprite's intricate lower structure. Evidence frames radio effects. Ionization grows around moving streamer tips, producing visible channels that divide repeatedly. This observed response measures ionospheric change. Different streamer polarities can develop in opposite directions within the same enormous event. This observation reveals halo development. Other events appear as simple luminous columns with little branching visible at ordinary video speed, within thin air. Sprites frequently occur in clusters distributed across a broad region above the parent storm. Observation records ionospheric change.
Successive sprites can appear to dance across the sky as lightning activity moves through the storm, across altitude.
Wind shear and tilted storm charge can shift sprites away from the parent ground stroke during brief flashes. The upper discharge may not sit directly above the point where lightning reaches the ground, above active storms.
A broad sprite halo sometimes appears before or without bright streamer development. This synchronized geometry reveals halo development.
Halos arise from diffuse electrical excitation rather than the narrow channels defining mature sprite streamers after powerful lightning.
Under suitable fields, a halo can transition into structured streamers that grow downward rapidly. Boundary locates ionospheric change.
Microsecond imaging reveals a developmental sequence hidden inside the brief visible flash. This measured sequence links streamer growth. Bright streamers can travel extraordinarily fast through the thin upper atmosphere. This field explains streamer growth above storms. The event extinguishes once local fields can no longer sustain ionization and energetic electron collisions within thin air. Although the light vanishes quickly, the upper atmosphere can remain electrically disturbed afterward. This sequence connects plasma motion.
Sprites and related events can produce measurable changes in very low frequency radio propagation. Emission exposes streamer growth.
Remote radio measurements help detect electrical disturbances even when clouds or distance block optical observations during brief flashes. Combining cameras, radio sensors, and lightning networks reveals how thunderstorms couple electrically with the upper atmosphere above them. Blue jets begin near storm tops and propagate upward through the stratosphere, rather than appearing suddenly far above. Unlike sprites, blue jets develop upward from the thunderstorm itself. This signal explains jet development within thin air. Typical blue jets terminate well below the highest sprite structures. This transition compares event rarity within thin air. Different colors reflect atmospheric density, composition, and the energy's exciting nitrogen molecules. This mapped profile connects charge transport. Blue jet emissions arise from excited and ionized nitrogen within denser stratospheric air above thunderstorm cloud tops. Brine streamers combine into the luminous structure recorded by sensitive high-speed cameras, revealing jet development frame by frame. Observed blue jets follow extremely brief electrical activity embedded within the cloud. This profile links plasma structure. Storm charge arrangement determines whether breakdown remains internal or escapes toward the stratosphere. This pattern tests global circuitry. Some upward discharges grow beyond ordinary blue jet altitudes and enter a different category. Response measures cloud escape. Gigantic jets can create a direct electrical connection between thunderstorms and the lower ionosphere. Profile connects jet development. Their upper branches reach approximately the same height as the base of the ionosphere. Pattern distinguishes event rarity. A single gigantic jet can extend many times higher than the storm producing it. Contrast tracks plasma structure. High-speed observations show that gigantic jets can advance through repeated leader steps and streamer bursts. Across altitude, different plasma structures coexist within the same upward discharge. This response constrains cloud escape within the upper atmosphere. Most attempted branches terminate before reaching the ionosphere. This profile links jet development within the upper atmosphere. The discharge changes structure as air density and conductivity vary with altitude. This mapped evidence frames charge transport.
Observed gigantic jets can transfer substantial electrical charge directly upward. This response measures global circuitry within thin air.
Gigantic jets represent one visible pathway connecting storm electricity with the planet-wide atmospheric circuit across wide regions. These jets are uncommon relative to the enormous number of lightning flashes occurring worldwide. Trace constrains global circuitry. Limited observations still prevent a complete global climatology of gigantic jets, especially across remote tropical oceans and continents. Elves are enormous, faint rings produced when a lightning electromagnetic pulse strikes the lower ionosphere, within thin air. An ELVE can spread across a region hundreds of kilometers wide within a fraction of a millisecond. The parent lightning does not need to send a physical channel into the ring. Measurement maps historical discovery.
Electromagnetic energy briefly excites the lower ionosphere and creates the expanding optical emission. This observation reveals historical discovery.
Its geometry reflects the outward-moving electromagnetic pulse intersecting a thin atmospheric layer. This timing exposes sensor fusion. Most ELVs vanish within about a millisecond, making them among the fastest transient luminous events within thin air.
Different event families occupy distinct timescales despite all being associated with thunderstorms. This synchronized boundary locates optical detection.
Photometers aboard spacecraft are especially effective at detecting ELVs. This sequence links sensor fusion within thin air. Viewing from above removes cloud obstruction and reveals the event's true horizontal scale. This contrast frames ELV expansion. Repeated orbital observations help estimate occurrence rates beyond what ground cameras can measure. This boundary maps historical discovery.
The first confirmed sprite recording appeared accidentally during an unrelated nighttime camera test in July 1989.
Repeated viewing revealed that the strange flash was real rather than a camera artifact. Contrast tracks event timing.
Shuttle observations soon provided more examples and confirmed that the phenomenon extended across large storm systems, across altitude. Different perspectives revealed structures that looked confusing or incomplete from the ground. This brief sequence links event timing. Coordinated observations reconstruct altitude, size, and timing more accurately than any single camera. Researchers combine optical imagery with storm structure and electrical measurements to identify triggering conditions. Network supports sensor fusion.
Millisecond synchronization separates plausible parent flashes from unrelated lightning occurring nearby. This structure supports ELV expansion above storms.
Faster cameras expose plasma motion that ordinary video compresses into one featureless flash. This channel distinguishes optical detection. Spectroscopy identifies which molecular transitions generate each event's characteristic light. This pattern distinguishes historical discovery within thin air. Color and spectrum provide remote clues about otherwise inaccessible upper atmospheric conditions. This map structure identifies event timing.
Space-based instruments now watch thunderstorms from above, where cloud tops cannot hide upward electrical activity after lightning.
ASIM combines imaging, fast light measurements, and high-energy detectors during repeated passes over active storms, above storms.
Orbital measurements captured blue jets beginning with extremely brief electrical activity inside cloud tops. Measurement maps forecast skill. Multi-channel observations distinguish genuine events from reflections, cosmic rays, and ordinary lightning glare. Field resolves ionospheric response.
From orbit, ELVs appear as complete rings rather than partial arcs hidden near the horizon, above active storms.
Astronaut photography complements automated instruments by targeting storms selected from forecasts and real-time observation. Within thin air,
Successful observations require predicting where intense storms will intersect a spacecraft's narrow viewing window. Observation records remaining uncertainty. Skilled citizen observers now contribute valuable images, timestamps, and storm locations to scientific databases. Timing clarifies sensor coordination. Multiple viewpoints can estimate altitude and structure, even when no dedicated instrument observed the event, during brief flashes.
reconstructions reveal how upward leaders develop inside storms before escaping into the atmosphere. Profile connects ionospheric response.
Scientists investigate whether transient luminous events measurably alter chemistry throughout the middle atmosphere and lower ionosphere locally.
Individual events are dramatic visually, but their global chemical importance remains uncertain. This mapped emission exposes ionospheric response.
Electrical disturbances can outlast visible light and temporarily modify the lower ionosphere. This resolved trace constrains chemical effects.
Background atmospheric waves may influence where breakdown begins and how sprite structures develop. This geometry records remaining uncertainty. Upper atmosphere conditions change continuously, complicating prediction from lightning measurements alone. This field explains orbital monitoring above storms.
Future systems may combine charge transfer, storm geometry, and ionospheric conditions to forecast events. Network supports ionospheric response.
Many questions were made about why apparently similar thunderstorms produce entirely different upper atmospheric discharges. Emission exposes orbital monitoring. The field has moved from disputed sightings toward detailed physics, yet important parts of the system remain unexplained.
Thunderstorms do not end at the cloud top. Their electrical influence can reach the boundary between atmosphere and space.
This South American fish can activate another animal's muscles without touching it, using electricity generated inside its own body.
Electric eels inhabit slow rivers, floodplains, and seasonally changing waters across northern South America, especially within quiet backwaters.
Their habitats are dark, cluttered, and difficult to search visually, making electrical sensing far more useful than eyesight.
Electric sensing turns murky water into a readable electrical landscape. The advantage matters during dark water navigation.
Despite its common name, an electric eel is a South American knifefish rather than a true eel.
A long anal fin provides smooth forward, backward, and hovering movement, while the elongated body remains nearly straight. Precise hovering keeps the sensory field aimed steadily, while the animal investigates prey hidden among roots and leaf litter.
Electric eels regularly reach the surface because atmospheric air supplies most of the oxygen supporting their metabolism.
A highly vascular mouth lining absorbs oxygen after every surface gulp. Control strengthens during fast predator escape. Air breathing permits survival in stagnant pools, where dissolved oxygen can fall below levels tolerated by many fishes. Most of the elongated body behind the vital organs contains specialized tissues devoted to generating electrical discharges.
Electrocytes are specialized cells arranged so their tiny voltages add together. That helps during muscle cell transformation.
Evolution transformed muscle-derived cells by reducing contraction, expanding membrane specialization, and coordinating their electrical activation together. Asymmetric nerve input lets each electrocyte create a directional voltage difference. Control strengthens during synchronized neural firing. A neural command opens ion channels almost simultaneously across the organ. Efficiency rises during sodium channel opening. Each activated cell contributes only a small electrical potential. Thousands combine to create the organ's full voltage.
Series arrangement adds the tiny voltage from each cell, producing a much larger discharge across the complete organ.
Parallel arrangement increases available current, while series arrangement raises total voltage. That helps during parallel current amplification.
Surrounding water completes the external circuit whenever a high voltage pulse travels from the eel toward another animal.
Genetic and anatomical evidence now separates electric eels into 3 living species, notably during species-level divergence. Each recognized species occupies a different combination of regional geology, water conductivity, and evolutionary history across Greater Amazonia. Electrical performance changes with the conductivity of surrounding water. Conductivity reshapes both current pathways and target exposure. Water chemistry reshapes the current pathway, changing how strongly a discharge crosses prey, predators, and surrounding habitat. Voltage alone cannot describe the biological effect of an electric discharge. Performance improves during multi-factor electrical effects.
Current, pulse duration, conductivity, and contact geometry together determine the biological effect of any electrical discharge underwater.
During a powerful pulse, the eel's head and tail behave as opposite electrical poles within the surrounding water.
Body position lets the animal shape where the strongest current travels. Precision increases during body position control.
Weaker pulses serve continuous sensing and communication, while powerful volleys are reserved mainly for predation and defense.
Objects with different conductivity distort the eel's self-generated field, allowing nearby structures to be detected without vision. Electroreceptors distributed across the skin sample tiny field changes created by nearby objects, prey, and environmental boundaries.
Electrical distortions become spatial information inside the nervous system. That neural map guides movement through cluttered darkness.
Active electrolocation reveals obstacles even when vision contributes almost nothing. The advantage matters during obstacle shape reconstruction. Living prey produces a distinctive electrical signature compared with stones, wood, sediment, and empty surrounding water nearby. Movement changes the pattern repeatedly, helping the eel separate prey from static background clutter during prey tracking. Pulse timing can change with context, arousal, and nearby electrical information. That helps during context-dependent pulsing. Weak electric discharges also carry social information between individuals without triggering a predatory or defensive attack underwater. Digital signals can help distinguish species, individuals, and behavioral states. Performance improves markedly during species signal recognition.
After detecting prey, the electrical system rapidly shifts from environmental mapping toward high-voltage muscular control instantly.
A hunting eel can activate nearby prey motor neurons remotely, forcing muscles to respond before physical contact occurs.
The prey's own nervous system carries the eel's externally imposed command. This matters during prey nerve firing.
High-frequency volleys force simultaneous contraction across the prey's body, producing a brief period of whole-body muscular tetanus. That temporary tetanus blocks coordinated escape during the eel's final approach through cluttered, low-visibility habitat underwater.
Controlled experiments reproduce the same muscular response by applying eel-like electrical stimulation directly to isolated prey preparations.
Closely spaced pulses prevent full relaxation between twitches, merging separate contractions into one sustained, forceful muscular response.
The prey becomes immobilized because its own nerves and muscles obey externally imposed electrical stimulation from the eel.
Electricity does not physically push the target. Instead, it recruits the target's motor neurons and muscle fibers.
External voltage crosses the threshold needed to initiate action potentials. That becomes valuable during motor threshold crossing. Neural activation closes the timing gap between detection and capture. Control remains useful during rapid neural conduction.
A sudden high-voltage burst can interrupt an escape turn before the prey completes the movement or accelerates away. Immobilization creates a short capture window, allowing the eel to close distance before coordinated swimming can resume.
Curling positions both electrical poles near the target. This bioelectric mechanism remains important during opposite pole positioning.
This posture greatly increases current through the prey's body. Curling therefore amplifies stimulation without raising internal output.
Curling strengthens stimulation through geometry, increasing current across the prey without raising the organ's internal voltage further.
Repeated volleys keep struggling prey from regaining coordinated movement. Continued stimulation preserves the eel's brief capture advantage. High-voltage hunting carries a metabolic and electrical cost. Recovery between attacks restores ion gradients across electrocyte membranes.
Between attacks, membrane pumps rebuild sodium and potassium gradients across thousands of electrocytes inside the electric organs.
Recharge depends on active transport, resetting thousands of biological battery cells. This matters during ion pump resetting. When prey remains hidden, the eel emits brief electrical doublets that can force an involuntary muscle twitch from concealment. That twitch moves leaves, sediment, or water, revealing the hidden animal against an otherwise motionless underwater background.
Laboratory doublets reproduced the same rapid muscular twitch observed when eels searched naturally for concealed prey underwater. The spacing between two pulses is well suited to summating the prey's muscle response into one detectable movement.
After delivering a doublet, the predator pauses briefly and monitors the surrounding electric field for nearby motion. Electrolocation converts the involuntary twitch into a precise positional clue. This remains important during twitch signal detection. Multiple viewpoints improve localization inside cluttered cover. That electrical strategy provides major advantages during multi-angle localization. Movement distinguishes living prey from inert debris more reliably than static electrical shape within cluttered underwater habitat.
Once the target is located, the eel immediately replaces probing doublets with an immobilizing high-voltage volley.
The same bioelectric system first exposes concealed prey, then prevents that prey from escaping the approaching predator. A complete hunt combines weak electrolocation, forced twitching, high-voltage immobilization, body curling, and rapid physical capture.
Low-voltage discharges continuously refresh the eel's environmental map before any predatory strike becomes necessary inside cluttered water.
Initial field distortion suggests a possible living target, but cannot confirm identity without additional testing underwater. A doublet challenges the hidden object to respond biologically. Any twitch separates living prey from motionless environmental clutter. The resulting motion confirms that excitable tissue is present. The eel immediately converts confirmation into precise targeting. Twitch geometry helps aim the following high-voltage strike. A clearer orientation strengthens the following immobilizing volley.
The attack volley recruits the prey's own motor system, turning ordinary muscle physiology into a temporary restraint.
Electrical control reduces the need for a prolonged physical chase. This remains important during rapid target capture.
Bioelectric hunting becomes especially valuable where darkness, roots, and sediment make ordinary visual pursuits slow or unreliable.
After feeding, the animal returns to low-voltage sensing rather than maintaining energetically costly attack volleys afterward. Electric eels also direct powerful volleys toward large nearby threats that cannot be treated as ordinary prey. During shallow water defense, an eel may approach vertically and rise toward the intruder instead of retreating. Pressing the chin against a conductive target creates a more effective pathway for electrical current through the threat.
As the eel climbs higher from the water, measured current entering the contacted object rises progressively and dramatically.
Emerging from the water removes competing current pathways, concentrating more of the discharge through the contacted threat. Repeated high-voltage volleys during brief contact can persuade an approaching predator or intruder to withdraw rapidly. Alexander von Humboldt famously described electric eels rising against horses driven into shallow South American water during an early expedition. Modern experiments confirm defensive leaping, although they cannot verify every dramatic detail preserved in Humboldt's 19th century account.
Instrumented trials confirm that climbing contact increases electrical delivery. Measured current rose as the eel climbed higher.
The same electric tissues power both predatory strikes and defensive leaps. That helps during shared organ deployment.
Sax organ contributes weaker pulses used primarily for electrolocation, orientation, and communication rather than forceful attack underwater.
Proteomic comparisons reveal distinct molecular profiles across the main, hunters, and sax electric organs within specialized tissues. Differences in channels, pumps, and structural proteins help each electric organ meet distinct energetic and signaling demands. Electric organs originate from muscle lineage cells during development. That process repurposes muscle precursors into non-contractile specialized electrical cells. During development, muscle precursors lose ordinary contractile machinery and gain the membrane specialization needed for electrical output. Electric organs evolved independently multiple times among fishes. Distant lineages repeatedly modified muscle through similar genetic pathways. Distant electric fish lineages repeatedly reused related genetic pathways while independently transforming muscle into electricity-producing tissue. Engineers examine electrocyte stacking for ideas about soft ion-based power sources, sensors, and bio-inspired materials today.
Bio-inspired research does not mean a living eel functions like a practical battery for ordinary human technology. Volta's electric eels have been observed gathering in groups exceeding 100 individuals near a remote Amazonian lake.
Coordinated movement herds prey into a dense, shallow aggregation. Herding concentrates many targets within one strike zone. Most eels maintain the encircling formation, while a smaller subgroup advances toward the compressed prey school together.
Several hunters then discharge high-voltage volleys nearly simultaneously. Timing several discharges together broadens the affected prey area.
Synchronized volleys can expose, stun, and scatter many small fish concentrated inside the shallow prey aggregations simultaneously. Nearby group members exploit the brief feeding opportunity created after several hunters discharge electricity together during feeding. The sequence can repeat during a single hunting event. Repeated cycles increase feeding efficiency under exceptional conditions. Similar events recorded in different years suggest structured social predation rather than one accidental gathering over time. Local prey density may make cooperative hunting worthwhile. Dense seasonal shoals can reward brief temporary social coordination.
Cooperative hunting likely appears only where prey density, shallow water, and local geography make coordination worthwhile for multiple hunters.
Group hunting is documented in Volta's electric eel, but should not be generalized across every species, habitat, or population.
Researchers still do not know how individuals coordinate each strike. The advantage matters during group coordination mechanisms.
Weak electrical pulses, body movements, and spatial position remain plausible signals for coordinating group attacks underwater together.
Field electrode arrays could separate individual discharge patterns during group hunts. This matters during field electrode monitoring.
Synchronized measurements may reveal whether attacks are actively coordinated. Combined data can expose timing of individual roles.
Protecting electric eel behavior requires conserving connected floodplains, tributaries, and seasonal channels throughout the greater Amazon basin.
Dams, pollution, and altered flood cycles can disrupt prey movements, breeding habitat, and seasonal hunting opportunities locally.
Healthy river connectivity preserves both electric eel diversity and the ecological conditions supporting their specialized behavior regionally. Together, electrolocation, remote muscle control, hidden prey detection, curling, leaping, and social hunting reveal exceptionally precise bioelectric behavior. Electric eels are living batteries that sense, command, defend, and sometimes hunt together through carefully controlled bioelectricity. The Great Pyramid still contains spaces whose purpose and exact shape remain unknown.
Khufu's monument belongs to a much larger royal landscape at Giza. Originally, polished limestone covered the stepped core visible today. Erosion and stone removal changed the monument's outer profile. Its builders established an exceptionally regular base.